All right, so my name is Rob Dempski, and I'm in the Department of Chemistry and Biochemistry here at WPI. And to start off, I know that we all are very engaged about what we want to eat and what we bring in our bodies, but sometimes we forget about what we actually happens after it gets inside. So I just wanted to start it with us, right, us in the beginning. And when we think about us, we're made of different organelles like hearts and kidneys and things where these nutrients must get to. And then we think about cells, right? All these cells are making up these different organs and organelles. And in fact, cells are, to a certain extent, they make up different organelles, but let's think of these cells as made of balloons, right? Because once we blow up the balloon and we tie it off, it's really hard to get something into these cells in the absence of popping them. And so our cells are kind of the same way. The normal cell layer, it can take a very long time for nutrients such as ions or sugars to be able to get inside of them. And in fact, in the absence of proteins, if there's a gradient across that cell membrane from the inside and outside, it can take over 100 years for it to equilibrate. And I think we would all agree that we would like the things get into our cells in less than 100 years. And so as we think about this, I was thinking, when I started my lab here at WPI, I thought about what kind of nutrients can I start to think about. And I started to hone in on these first row transition metals. And that includes iron and zinc and copper. But in fact, to pick one of those is right here is zinc. That's what I chose to focus. How does zinc get into cells in our body after we eat all of those foods? And in fact, zinc is important all throughout the body lifespan. So when you are a baby and you have your mother who's feeding you as a baby, there's zinc levels increase dramatically into the cells, so the babies can get lots of zinc. And what has been shown is there's a small number of babies that once they're weaned from their mother's milk, that they get these rashes and they get this disease, and it causes neuronal problems, growth retardation. And so in fact, zinc is really critical for a homeostasis. And as we go along through life, zinc is really important, certainly in the developing world, because zinc deficiency ends up causing gross abnormalities in cells. And so as we sit here in Massachusetts, we may think that this is not such a big deal in the US, but in fact, as we get older, there are a number of disease states which are important when you have alterations in zinc. And this includes Alzheimer's. It also includes pancreatic cancer. So in 2019, there's about 55,000 people will be diagnosed with pancreatic cancer, and there will be 45,000 deaths directly related to pancreatic cancer. And in fact, the proteins that we study in our lab have been shown to both initiate and progress pancreatic cancer. So therefore, we thought this was an interesting target to look at the proteins and how they function. But when I opened my lab in 2009, this picture up here is essentially the high-resolution structure of these ZIP transporters, or zinc transporters. And in fact, what we can say is we really don't know very much about it. There's a couple boxes, there's a couple loops. We know it goes on the surface of those cells. But other than that, we really don't have an idea of how to look at these proteins. So through a combination of experimental and computational approaches, we were able to devise a structure. And in fact, so this is what our model is of the protein that studies that imports zinc into cells. And if we expand upon that silver part, which is in the middle, what I think we can all agree is that the middle of that protein structure looks much better than how we started in 2009. And so now we can start thinking about how does this protein function and how does it function in these disease states. Now, furthermore, what happened earlier this year is we showed that this loop on the bottom, which is essential for maintaining zinc in cells, is disordered. In other words, it has no structure. It just kind of flaps around like cooked spaghetti noodles. But what's critical about this is that those spaghetti noodles, in fact, they regulate zinc in the cell, and they also, dysfunction of this domain results in disease states. So our question is, how does that domain influence order in the cell? Now also this year, as we move from computational to experimental approaches, we've now gone into cells. And so this is just a cover that we had in a journal earlier this year. And what we see is mammalian cells, which is in the background, and then our model, that same model that I showed before. And now what we can do is we can quantify the oligomerization of the cell on the surface of cells, and we can see how that influences both healthy and disease states. And so we're really excited about this because we think we can answer sort of fundamental questions, such as how do ZIPs influence health and disease states? How does disorder on that little loop induce order? As well as can we start thinking about therapeutics to be able to treat diseases like Alzheimer's and pancreatic cancer? And of course, I wouldn't be able to do this without superb coworkers here at WPI, as well as collaborators who are across the US, and funding from this institution and the NIH. Thank you.